So what I'm going to do is I'm just going to run through a couple more examples of, getting the feel for how we implement these tests and what kind of scenarios we might look at. So the next scenarios that I'm going to set up with are, for example, saying something about we're looking at sea snake populations, if I could actually talk. We're wondering about how they're going to, be impacted by temperature changes. Could this be positive? Could this be negative? Will it change the distribution of where they're going to be found? All of these kinds of things are things that are going to be playing out when we have climate change running through the system. So we might need to think about where we're going to put our conservation areas. Do we need to be thinking of shifting them? Or are our, all of those kinds of things are going to be at play. You can also imagine that it could be quite difficult to get ethics approval or collect rare and endangered snakes. So by their very nature, we're going to have a small sample of these. So we might be thinking that, small samples, we're a bit worried about what kind of underlying population distribution they have there. Maybe this is the case for thinking about non-parametric tests. And this is, realistically the kinds of decisions people that want to undertake research in this area are going to be faced with. So we've got some hard to collect data. What we want to do is we want to move what we've collected. We have some observations in tanks, some in wild. So, getting the wild ones very hard to collect. So really what we're trying to set up here is saying, are they going to behave the same in our tank setup as they do in the wild? And if they behave the same in our tank setup, cage, tank, we use a generic term cage, but in this case, obviously it would be a tank. If they behave the same, then we might think that we could do some more research with the sample that we have in our caged environment. If they're behaving differently in the wild to how they're behaving in captivity, then we might feel very, very much less confident in trying to use, experimental conditions to try to make any inferences about how they might behave, as things change. So we might be interested in seeing how they respond to different temperatures, but before we, sort of get that far, we think we want to understand, are they going to behave the same way when we put them in the wild as they do in tanks? So we might have set up some, cameras underwater to be collecting this kind of information, sampling for, 24-hour periods. This is quite, expensive and difficult stuff to do, but we've got some data, or at least someone in our research team has been able to get some wild observed data. And so we've got our guys in the tank. We're going to then do another experiment. We're going to videotape them in the tank, and we're going to see how much they move, get some measurements on their movement, and then see if we can compare the two. If the caged animals are as active as our animals in the wild, then we might think that, okay, we can start to make some inferences from those that we have in the lab. Okay, so small samples, again, it was very hard to get that wild data. So here are our movement measurements. So we have 8 in the cage, and we have 5 observations out in the wild. So this is our standardized measure of movement that we've captured. So we've got eight different snakes that we've got in tanks in the lab, and we observed 5 different snakes in the wild. So these are not repeat measures on the same snake, okay? So that's why it was so hard to get them, right? So very, very hard to get when there's a rare and endangered species, perhaps. We've got some in captivity, perhaps they're from a breeding program. We want to know, are their movements the same? What is going to be the process that we use? Okay, same process that you're getting very familiar with. We're going to create a box plot. We are then going to formalize a null and an alternate. Okay, body movement intensity, the same in the wild as that of captive snakes. Body movement intensity, different between wild snakes and captive snakes. And the reason we want to know that, remember, is that we want to be able to, understand whether we can use the captive snakes to draw inferences probably about the way maybe movement changes with temperature, that kind of thing. Okay, we're going to implement our non-parametric test in R, and we're going to interpret the p-values. So there's our plot. Okay, you can see that the two outliers are identified there, so that's something to not be frightened of when you look at your samples. So you can see that there are no whiskers here. This is just applying the formula, okay? So it just means that the interquartile range also happens to fall at this point. There's no further observations outside that, just our end points, okay? And that's because we only have 5 data points really. So don't worry about when you have small sort of samples, you can get this sort of result. For our cage guys where we had 8, we got something that looks a little bit more like what we're normally prepared to. But when you have small samples, don't think that you've done anything wrong. You've seen an example here. So we have no whiskers on this plot. We just have 2 outliers and the interquartile range and the median sitting right there. And that's a possible outcome when you have a small data set. So definitely don't worry about that. We want to conduct the test. So we need to read in our data. So I give it a name, snake A, caged, wild, comparison. That was my data file. You can see I've got my movement intensity data here, and I've got my factor variable in here, whether they're in the cage, in the tank, or whether they're in the wild. I only had 13 observations. How do I implement my test? I just want to have with the snake A data, Wilcox.test, movement, tilde, environment, which are the names of my variables. And you can see that the p-value is above 0.05. So I would say I have insufficient evidence to conclude that the movement intensity of the captive snakes is different to those of my wild sea snakes. And that's helpful for me because it's very hard to get, those observations on those wild sea snakes for, practical reasons. So now that I've established that, the research group is moving on, it's doing its next round of tests, or the next lot of honours students have come along and they're looking for their, research program to do. So what might their little experiment be? Well, now that we are confident that we can make inferences about the wild population from our captive population, at least in the direction of movement intensity, we might set up a test. So this is now an experiment, so we're going to have normal temperature, and we're worried about warming temperatures, so we're going to crank it up. And this is, a non-trivial difference. So remember, small differences, it's going to be hard to detect a difference. We're going to have low power. So we've got a relatively small sample of captive snakes. So, we're going to look for setting the difference quite large, observe over a 24-hour period, we're going to conduct our non-parametric test. So here is our movement. So we had 8 captive guys in normal temperature. We then going to have those same guys at 2 degrees higher. So remember, I've got my 8 captive sea snakes, so I've got 2 measurements on each snake. One at the normal temperature and what their movement intensity was at the higher temperature. Okay, so paired, I've got 2 measurements on each snake. I've only got 8 snakes in my captive breeding program, so I put them all. We have them in the normal temperature, run the camera, get our standardized measure of movement intensity. I then, wait a week, crank up the temperature by 2 degrees, run the experiment for 24 hours, record against each tank movement intensity again. So I have 2 measurements on each sea snake. That's my experimental design. So I'm going to have my plot, which is going to be a horizontal box plot. I need to formalize my null and my alternate, which is movement intensity is the same under normal temperature conditions and at plus 2 degrees. Set my alpha. Where does that come into play? It comes into play when I'm going to apply my test to decide whether or not I'm going to reject the null. So p-value decision rule, we're going to reach a conclusion. So here is what I get when I get my paired t-test. So I plot this on my data scale. Remember, my test is going to use the ranks of the data, but we plot it on the original scale. You can see that there looks like there's some more movement happening on here. It's not 100% convincing, but I would say that generally these guys look like, there's more movement when we have warmer temperature, I'd be thinking there's a difference, but we will conduct our formal test and see what we get. So I've read in my data here, I've read in my data, this was my CSV data file, this is the name I give it in R. I conduct my little test to see what's in my data, so this is the heading, so I've got in the normal and high, whatever you might use to denote your column headings. We conduct the test, we've seen how to do this, pretty straightforward. With snakes, Wilcox.test, paired is equal to true. Look at the p-value. P-value is less than 0.05, therefore we reject the null. And you can see here that we got some paired values, but they bite at the third or fourth decimal place, or even further down. We make our decision rule at the second decimal place, so we don't have to worry about that. So we've seen some examples of paired non-parametric tests and an non-paired non-parametric test. Sorry, there are 4 possible combinations. I think I've just confused myself, so I'll move on and give you a summary. So whenever we have a parametric test, we have a non-parametric equivalent. We use the non-parametric equivalent when the assumptions of the parametric model don't hold. These are usually when we have small samples and we have strong evidence that the population is not normal. Remember, if we had a large sample, we wouldn't care what the shape of the population was because we could appeal to the central limit theorem. Our practical steps that we know what we're going to do. Parametric tests we're going to implement with the same plot as we have for our t-tests and everything else that we've done to date, so that's easy. Our null is a slightly easier version. We don't test the means because our tests rely on the rank order of the data. So we just have these very general statements about the null and the alternative. A is the same as B. A is not the same as B. It's pretty straightforward. We're always going to have our alpha at 0.05. So that's easy for us to apply our decision rule. It's very helpful because the language in this area is quite confusing. And we're going to have implementations in R, which are really easy. You've got 2 ways of having your data in R. You can have it in what's called long format. When you have it in long format, you have length tilde type. When you have it in wide format, you have it with a comma, okay? And so if in here, if we want to have a paired test, we just put the paired equals true. If we want a normal, not paired test, we just don't put anything. We look at these p-values. If they're small or less than 0.05, we reject the null. We're going to see for these tests, a lot of these warning messages come up, and that's fine because we're just in a position to ignore those warnings. And that, again, is an opportune time for a break.